All right, thanks everybody for coming this morning. It's always very exciting when a student graduates. And so, yeah, Grace will be coming up soon to tell you about what she did for her thesis. I'm first going to do a little bit of introduction. For people that are online, at the end of the defense, you're welcome to write questions in the comments section, and we will read those out to Grace. So if you want to make her squirm, you can leave her a few comments there. And yep, thanks to Grace's family that's coming. Her dad Paul is here, her mom Julia. And any other family members?
No.
Online maybe?
Online, yes.
Nice. And I'd like to thank Grace's committee, so Cheryl Logan and Tom Connolly. And so let's get going. Let's give you a little bit of an intro for Grace and kind of her pathway here to finishing her master's. So, there's the real Grace. A little sillier photo. Grace from the beginning, she's living her best beach life, right? On her path to being a marine biologist, even growing up, spending time with her family, burying in the sand, taking naps, strutting down the beach, right? There are some early signs that she was on this pathway to being a marine biologist, playing in tide pools, I assume looking for little fishes, right? Catching fish. She's looking a little apprehensive here about what do I do with this thing, right? Maybe even bigger fish, right? Even bigger fish. And then, well, that's not a fish, but she's definitely looking a lot more confident holding that toothy creature there. Right? Grace, even from an early age, right, she learned some really important lessons about the food chain, right? So about 25 years ago, Grace's point of view, little toothy thing gets eaten by big toothy thing, which that actually kind of happened when we were out doing some fishing for some cabezon for Alex Stella's thesis, and we caught this big lingcod. What this photo doesn't show you is that a few moments after this, Grace ended up being the little thing inside the mouth of this big thing. She didn't lose too much blood, right?
Just feeling in my thumb.
Yeah, just nerve endings severed by those sharp teeth. Right. Grace definitely enjoys spending time in nature, especially with her family. Right? And I say mostly here because there's this lovely image. I think we've all felt that way on a backpacking trip and not showering for multiple days, and that expression is priceless. Grace is also, she's in sports, she's very athletic. Had some great photos of her. She shreds, put her on some waves, put her on some skis, there with another group of Moss Landing students. And then this, whatever this is, this new sport of backyard boogie boarding with your cat. Grace is also an accomplished artist. At least I assume your mom sent this picture. I assume that's stuff you did. Is that correct?
Yeah.
Amazing. Look at how amazing she is. Painting, and then her poetry in college was featured at an exhibit in the Santa Barbara Library. Grace also appreciates cooking and, good meals with friends. Even when she was young, she cooks up some spectacular meals, especially while camping. Opening the can of the finest caviar, probably, right? Yeah. This lovely meal at camping. This is my favorite. Double fisting on the couch. Grace had a bit of work experience. So before coming here, she worked for the California Department of Fish and Wildlife as a scientific aide for their California Recreational Fisheries Survey. And went there, she would go around to the ports and sample the catch of the fishermen and talk to them about their catch and measure things. And she said, she put this in her application to Moss Landing. She said, before beginning my current job at Surfs, I often fantasize about working with creatures a bit more charismatic than rockfish. So I ask you, that's what she chose to study. Am I more charismatic? We'll see by the end of her thesis. And then also while she was here in Moss Landing, she spent many years working at Lemongrass, the local Thai restaurant, and I know they were kind of a second family to her there. And if you haven't eaten there, it's some of the best food in the area by far. A little bit about Grace's academic journey. So she did her undergraduate degree at Westmont College in Santa Barbara. She majored in biology, ecology, evolution, and natural history. She graduated summa cum laude. I think she had her worst grade was like an A minus maybe. And I looked back at the recommendations from all her teachers there, and they just, they were all saying Grace is like one of the best students we've had in decades. Pretty much anything that she does, I'd have to agree. At Moss Landing, she did fantastic in her classes. She was a teaching assistant for 5 classes. That might be a record, and I think it's because she did such a good job. She was a TA for my marine ecology class during the infamous COVID year. She was a TA 3 years for scientific writing, and she's really a fantastic writer. Everyone who's a student of mine knows that I love to write a lot of comments on their writing. And for Grace, she would turn it in, and I'd be like, man, even the first draft, I was kind of searching sometimes to leave her comments because she's such a good writer. She was a TA for biological oceanography. She got some funding for her research from CSU Coast and the MLML Wave Award. And then the first conference she ever went to present on her research was the California Estuarine Research Society meeting, and she won Best Student Talk. So, all that academic excellence certainly can be tiring. And so now Grace is going to come up. She's going to tell you about the work she did for her thesis, which was studying the effects of hypoxia and warming temperatures on the nursery function for these 2 flatfish species, English sole and speckled sand dabs. She'll tell you all about the trials and tribulations of doing caging in the slough. And, Grace was a fantastic student, and, very meticulous, kind of a perfectionist, but always did just fantastic work. And I'm really excited for her to be here. And here's another quote from her application to Moss Landing. Many students, there's what they say they want to do, and there's what they actually did do, and they usually are not the same. But for Grace, she said, as a student, I would like to study the impacts of various environmental factors on the status of fish species. I wish to explore the effects of climate change, ocean acidification, disturbance, pollution, and natural oceanic processes on species of commercial and recreational interest. Further, I am interested in investigating factors that might influence species-specific reactions to these elements. That's pretty much exactly what she did. Climate change impacts on fish, pollution from eutrophication in the estuary, natural processes in terms of these daily fluctuations in all these environmental conditions, and commercial and recreationally important species, and species-specific 2 different species comparisons. Did it exactly. Very nice job. Right? She also, maybe she's going to mention this or not, but she's also, was able to overcome adversity. One example here, there was an infamous time where the boat got a little too shallow, and the tide was going out, stuck in the mud, and Kinsey, who's in the audience, and Grace, valiantly tried to get the boat out. You see the after effects there of all the mud. And then, at one point, there was finally a rescue was organized, and they were saved. But, that's all kind of goes with the territory of doing work in the slough. So Grace is going to come up now and tell you about her research. And so let's all congratulate her just the way she would.
Thank you. All right, thank you, Scott, for that introduction, and everyone for being here for my thesis, Assessing Nursery Habitat Quality in Elkhorn Slough, Effects of Elevated Temperature and Hypoxia on 2 Juvenile Flatfish Species. So to begin, I'm going to talk to you a little bit about estuaries, these very unique, very productive habitats where the rivers transition into the sea. They provide us with a number of ecosystem services, including coastal recreation and protection, and they can act as carbon sinks, and they're also widely established as nurseries for juvenile fishes, which will be the focus today. So to be a nursery, it just means that you are contributing significantly to an eventual adult population of fish relative to other available habitats. These are places where juveniles tend to concentrate because they offer abundant food, refuge from predators. If you're a small little fish, other fish, their mouth isn't so big, so you're going to be targeted from gape-limited predation. So the faster you can reach a certain size, the less at risk you'll be. And then they'll also have conditions for rapid growth, because then again, once you reach that size, you'll be safer, and then you can migrate offshore as an adult. So compared to the open coast, estuaries tend to provide well in these categories, which is why juveniles tend to thrive there. But things are not always so perfect. So Estuaries can be extreme. They're extremely productive and biodiverse, but they can also undergo some extreme stressors. So a lot of estuaries are located by agricultural areas and urban areas, which means a lot of pollution, a lot of human products entering the system as runoff. And so that can lead to a problem called eutrophication, which is excess nutrients in the system. And although nutrients are vital, they can promote algal blooms, which are not only sometimes toxic, but as they decay, they can also deplete the water column of their oxygen. And then warming and hypersalinity can also coincide with these stressors, especially when you have shallow estuaries. So sometimes there's restricted tidal flow as well, and naturally these systems undergo a lot of flux on a daily and even an hourly scale. So any stressor that a fish is experiencing in the open ocean, whether that's acidification, hypoxia, or warming, that is going to be even worse in an estuary. And because of estuaries' dynamic nature and the various threats they face, it's important to monitor them so that we know whether or not they're staying suitable for sustaining the many lives that depend on these ecosystems. And in particular, water temperature and dissolved oxygen, or DO, are two parameters that strongly influence the well-being of juvenile fishes and therefore can help determine whether or not we have a good nursery site. So for instance, extreme heat and extreme hypoxia can result in mass mortalities or fish kills. And they can also do sublethal effects such as reduced growth. If it falls outside of a fish's thermal range or preferred oxygen level, they might also avoid areas that do that, which will force them into poorer quality habitats that either have less prey or more predators. So exactly when we start to see these problems can be hard to define at sometimes, and then it also varies species to species. So normal oxygen level is generally described as 8 milligrams per liter. And the Water Quality Control Board for the Central Coast region where we are says that healthy estuaries should not ever drop below 5 milligrams per liter. 4 milligrams is a common threshold you'll see because that's when a lot of fish start to experience sublethal effects like behavioral changes and reduced growth. And then 2 milligrams per liter is another definition for hypoxia that's common because it's lethal for a lot of species, and so when an estuary drops below 2 milligrams per liter, that's when you start to see biodiversity really start to drop. So our story today is set right here in Elkhorn Slough, one of the largest estuaries in the state, and a couple of things to note. Almost a quarter of the surrounding land is used for agricultural purposes, so that means terrestrial runoff and pollution can diminish the water quality. And it also experiences varying levels of eutrophication from moderate to hypereutrophic, especially in the upper reaches and those areas behind tidal gates. And then it experiences both seasonal and daily hypoxia patterns, especially in the summertime. That's when hypersalinity, warming, and hypoxia all tend to coincide with one another. But even so, Elkhorn Slough is teeming with life. It's a popular birding destination. It's home to a lot of key invertebrate populations like the Olympia oyster. It's a good place to spy marine mammals, and it's home to over 100 different kinds of fish, many of which rely on it for nursery habitat. So English sole up in the top right, over 50% of our local adult population starts out in Elkhorn Slough, despite the fact that geographically it only represents a small portion of available habitat. So of those many fish that it's home to, I'm going to talk to you about English sole and speckled sand dab. And both of these fish are abundant in the slough, and they use it primarily as a nursery ground. So they start there in the spring and summer, and then will generally migrate offshore by the end of their first year. But there are a few key differences. With English sole, live a lot longer and get a lot bigger. So they live for over 20 years and reach over 2 feet, whereas speckled sand dabs only live for a few years and in that time only reach a few inches. So because of that, English sole are targeted by the commercial trawling industry. Speckled sand dabs are too small to be a commercial target, but they do still contribute as an important recreational hook and line target. They also have different habitat preferences, so English sole tend to gravitate towards mudflats and eelgrass beds, and speckled sand dabs like shallow sandy habitat. So both of these species are shown to be pretty resilient to hypoxia, but previous research suggests that they will avoid regions of the slough that are hypoxic, and that the speckled sand dab may be the hardier of the two species. So this is from a 2015 study from Brent Hughes, and what you'll see with the graphs on the right is that speckled sand dabs are twice as more likely to appear in regions of 4 milligrams per liter of DO than English sole. And then on the lower left-hand side, that's showing you that when the estuary becomes hypoxic, both species are mainly just found in the lower parts of the slough where oxygen levels are higher. So with this study in mind, I wanted to mechanistically test whether DO and other factors impacted the fish at different sites in the slough. So now that we understand how Elkhorn Slough is important as a nursery and how these two fish use it and might use it differently, I wanted to explore the following questions. Do environmental conditions in the slough differ spatially or seasonally? So I ran 2 experiments, one in August 2021 and one in November of the same year. And then do growth and survivorship of these 2 species differ spatially or seasonally? And then finally, if so, which environmental factors are associated with those different growth and survivorship rates? And are there any notable thresholds constraining the growth or survivorship? So to answer these questions, I selected 9 sites throughout Elkhorn Slough to deploy the cages of the fish, and I monitored them for up to 3 weeks, as well as monitoring the environmental conditions. And the purpose of this cage study was to assess the nursery quality of different regions of the slough by exposing them to a variety of real-life combined stressors similar to what they would be experiencing in the wild, and see how they responded. So I'll walk you through the 5 main components of my methods. So that would be site selection, fish collection and preparation, cage building, field sampling, and data analysis. So as I mentioned before, Elkhorn Slough varies spatially with regards to eutrophication and overall water quality. So every year, ESNR, the Research Reserve Foundation, they collect volunteer water quality monitoring data, and they score the estuary from 0 to 100 based on how far each of those parameters deviates from normal or ideal conditions. And they score based on DO, dissolved nutrients, turbidity, and pH. So their monitoring sites are going to be the circles on the map. So as you can see, there's basically a gradient. It gets worse the farther up you go, and also is worse at tidally restricted sites behind artificial dams. I'll also mention here that they have some long-term monitoring sites where they measure water quality every 15 minutes, but only one of those was in the main channel where my experiments took place. So for the scope of this project, I'm not going to mention those data. And I selected sites only with full tidal range because a similarly designed study in 2016 found that behind the artificial dams, conditions were super hypoxic, and so their staghorn sculpin, which is another really hardy slough fish, died within a couple of days. And because I'd only be monitoring my cages once a week, I didn't want my fish to die that quickly, so I wanted full tidal range, but also expanding the full length of the slough to capture different conditions. And because this water quality report card is an accessible tool that is supposed to represent the current status of estuarine health in Elkhorn Slough, I used it as a guide to classify my selected sites as either poor or fair quality. So anything that they graded as an A or B, I considered a fair site. Anything lower than that, I considered a poor site. And now we're going to journey through the slough a little bit to talk about the differences between each of those regions. So the upper slough, it's considered poor according to the water quality report card. It's shallow, it's muddy, there are a lot of tube worms, low species richness, but still fish can and do access the upper reaches of the slough. There's high benthic invertebrate abundance. So even though it's low richness, there are a lot of prey that are available, so that may be a draw factor for fish to go into the upper slough. So I had 2 upper slough sites, Hudson's Landing North and South, And they're pretty close together, but a pilot study showed pretty marked differences between them in terms of fish growth and also oxygen and temperature. And then middle slough sites, also poor quality, also muddy, highly turbid. Fish assemblages, though, more similar to what we're seeing in the lower slough. So I had 3 sites total. Azevedo Pond, which, as a disclaimer, I'm not referring to the Azevedo Pond that you'll usually hear about, which is a really small hypereutrophic water body behind a dam. I'm calling that because it's in the main channel right next to that, and that's the nearest named landmark. And then Kirby Park. And Rubus Creek was selected in my November experiment to replace the Azevedo Pond site, just to try to capture conditions a little bit more intermediate between Kirby Park and the lower slough. And then so the lower slough is where we start to get into the fair sites. So these are a lot of sand flats and eelgrass beds, only moderately eutrophic, typically the most stable part of the slough environmentally. Higher DO levels, except when they decrease in the springtime from upwelling. High invertebrate species richness, but lower in abundance. And this is where we expect to see the best quality nursery sites. And I had 2 non-slough sites as well. So these were both connected to the main channel, but not really part of Elkhorn Slough proper. So I'll start with the harbor site, which I selected to kind of act as sort of a control site. Its conditions were gonna be most similar to what fish would experience on the open coast. So you're gonna have sandy bottom, high visibility, but lower invertebrate abundance in the benthos. And possibly in the wild, higher risk of predation here as well. And then the Old Salinas River Channel site, which is actually just right outside that window over there, this is considered a poor site because it's highly eutrophic. It's going to be influenced both by freshwater and saltwater input. Very muddy, very turbid, but invertebrate abundance is actually higher in this site than it is in the lower slough. So now that I have my sites picked out, we had to collect our fish. So the main collection method we used was otter trawling. We trawled the slough for about 10 minutes at a time at low speed. And then when before I put the fish in the cages, I had to be able to distinguish between them individually. So we used these visible elastomer tags, just a liquid gel that you can deposit beneath the skin, and then it'll harden into a non-toxic solid. And then to deploy the fish, I needed to build a bunch of these cages to house them in the estuary. So the purpose of the caging method was to hold the sites at specific sites where I could monitor the water quality, and so I could keep track of the same fish and find them every week and record their growth and survival rates. So that's a well-established method. And then in 2003, Jennifer Brown did a study that monitored their growth rates and found both speckled sand dabs and English sole grew well when they were caged in the estuary. So that means they're able to feed from within the cages. So PVC pipe was the skeleton, and then Vexar mesh is kind of a stiff but slightly flexible plastic material. It had holes that were big enough to allow water flow and access to the prey, but small enough to keep my fish in and then keep the predators out. And then we have the rebar and cement that kept them submerged in the mud so that they could really access the food. Floats so that we could find them and access them. And then one cage at every site had a mini dot sensor, so these recorded dissolved oxygen and temperature every 20 minutes. For the purpose of my data analysis, I took 24-hour averages of these measurements or used the raw data to calculate how long each site was spending below certain oxygen thresholds like 2, 4, and 5 milligrams per liter. So carried out 2 field experiments, one in August 2021 with English sole, and then another for speckled sand dab in November. Summer and fall are both typically considered what is part of Elkhorn Slough's dry season. So we took the fish out in a boat on a cooler and then deployed 2 to 3 cages at every site. Measured and weighed the fish at the beginning. So then during each cage check, we would measure the fish to get length, we would measure mortality, and then we would replace all of the dead fish with live fish to maintain consistent stocking density. And then we also took weekly measurements of pH and salinity at each site. So they stayed out there for a maximum of 3 weeks, but if at any point more than 80% mortality occurred, that site was terminated. And then just briefly, data analysis. So to remind everyone of my questions on the board, I was using mostly analysis of variance, principal component analysis, and regression to answer those questions, but we'll get into the details a little bit for the results. And so before talking about some figures, I'm just gonna tell you where we're about to go. So first, we're gonna take a look at all of the mini-dot data to answer question 1, whether or not there were daily, or seasonal, or spatial environmental trends in the slough. And then I'll show you how the fish actually did in terms of spatial variation. And then we'll look at take what we learned from question 1 and question 2 and use that with the principal component analysis to answer how and why environmental conditions might be impacting overall nursery function in the slough. And then finally, I'll share some challenges, recommendations, and broader impacts of this research. So first, we can talk about daily environmental patterns. So I mentioned this earlier but when providing background, but now we can actually see it with the data that I collected. So these are subsets of time series data that just show you 1 or 2 days. But what you can see is that every site, which is a different color, experienced different things, but they're all doing the same pattern where they're peaking during the daytime for temperature and oxygen when it's warm and sunny and there's a lot of photosynthesis happening, and then dropping at night when temperatures are cooler, but there's no photosynthesis, no oxygen production. It's just organisms respiring and using up the available oxygen. And so to expand a little bit on that, DO and temperature generally are inversely related. So that's a well-known concept with solubility. Warmer water just does not hold oxygen as well. So this is Hudson's Landing, and that just shows you that pattern where you have when temperature spikes a little bit, oxygen is dropping down. But at a smaller time scale, you can see where those spikes daily are actually coinciding. So there's a strong positive relationship between the two because, as we saw in the previous slide, biological processes are largely controlling that pattern, especially in the upper reaches of the slough, where they're not being flushed out as often. So these are the full time series plots for the mini-dot data. So we have August on the left and then November on the right. And so what I want you to see here is that there's a pretty distinct separation with how the groups are together for temperature during both experiments. We have in the red at the top, those are all the upper slough sites that are getting really warm. And then you can see it still for dissolved oxygen in August as well, where the lower slough sites are going to be lower, and then the upper slough sites have lower DO, and then it's reversed as you go down the estuary. And I wanted to show you the previous slide just to get an overall idea of the fluctuations of different sites over time. These graphs are showing you the same information, but now we can see the site-by-site separation a little bit better. So on the left with August, you can see that each site experienced different oxygen conditions. And for the most part, those fair and poor designations are holding up such that the fair sites are more oxygenated and then the poor sites are lower. Same thing with temperature. It kind of follows a pattern as warmer as you go up, which is where most of those poor sites are. Then in November, when conditions were milder, dissolved oxygen was basically the same across the board. Temperature followed the same pattern of increasing at poor sites, but overall was much lower than it was in August. And then one thing to note on the bottom left in that OSR, Old Salinas River Channel, even though it's a poor site, it's located closer to the lower slough, so it actually experienced temperatures that were closer in condition to the fair sites than to the poor sites, which will be important to remember later. And now looking at some of those calculations. So we're looking at 10th percentile DO, which accounts for both amount of time, frequency of hypoxic events, and also how low the oxygen was, and then 90th percentile temperature. So in August, significantly, the poor sites had a lower 10th percentile DO, and higher temperature. Then in November, there was no significant difference between poor and fair sites, but we do see like a little bit of a trend being slightly higher at the fair sites. And then again, temperature, it was warmer at the poor sites. And for each of those thresholds that we discussed before, 2, 4, and 5, in August, poor sites spent significantly more time beneath those critical oxygen thresholds. Then in November, no sites dropped below 2 milligrams per liter, and then they barely dropped below 4 and 5 as well. Again, slight trend towards more at the poor sites, but it wasn't significant. And now looking at the spot sampling for pH and salinity, there was a trend towards lower pH at poor sites. So those are sites generally located by freshwater input, and freshwater is slightly more acidic than ocean water, but there wasn't a significant difference for either month. Then in terms of salinity, for fair sites, which are closer to the ocean, those were lower in salinity in August than poor sites were. It wasn't significant, but if you take out those bottom outliers, which was Old Salinas River, had lower salinity. So excluding Salinas River, poor sites became really, really hypersaline. And then in November, that pattern reversed. So poor sites were less saline because it was a wetter season, so you had more freshwater input into those systems, and there was less evaporation, so they weren't getting as hypersaline. So to recap all of the environmental things, in August, poor sites were characterized by higher temperatures, lower DO, hypersalinity, and maybe lower pH. And then in November, still higher temperatures, but lower compared to August, lower salinity, and then also maybe lower pH. And so now let's look at how the fish did. They died, so I'll show mortality a couple of different ways. This is just plotting the proportion of surviving fish at each site. So we have sole on the left and then sand dabs on the right. And then each little individual point or triangle square that's representing the mortality rate within a single cage. And then going from left to right on the axis, you're moving from the harbor and then you're going upstream. So what you'll see is for Seoul, Kirby Park and beyond, the survivorship starts to go down, whereas the lower slough, they all survived really well. And then in sand dabs, the survivorship was overall higher, but same thing, like once you pass the middle slough, that's when you start to see more fish dying. And so these results do reflect a similar pattern to what we saw in the Hughes study earlier, which if you recall said that when you have periods of normal oxygen, the fish are using the whole slough, but then when you get into hypoxia, they're confined to that lower slough. So my results are suggesting that they're not only avoiding it because it's not ideal, but they might not even be able to survive there if they were to stay. And then these are Kaplan-Meier survival curves. So basically they record what mortalities occurred and didn't occur, and they can use that to estimate mortality survival rates over time. So again, sand dab survival was overall higher, but for both experiments, poor sites had a significantly higher mortality rate. So even though they were under different conditions, there was something going on at the poor sites that made them less conducive to survival in both instances. Moving on to growth, this slide will show you 3 different growth metrics. So we're looking at linear growth rate, which is a simple change in length over time, specific growth rate by length, which takes into account the percentage change. So we're measuring the same fish over and over, so that accounts for that. And then specific growth rate by mass, the same thing, only using the before and after weights instead of length. And what you'll see here is that Old Salinas River actually did very well and did the best out of both the fair and the poor sites. But generally, other than that, the middle slough and upper slough were worse for growth. And then you can especially see that in the last graph in terms of weight. Some of the weight data are missing because if fish died before the end of the experiment, we don't have a final weight for them, so we weren't able to calculate that. And if you remember from earlier, Old Salinas River had low oxygen levels, but actually also lower temperature and lower salinity, so that may have been why why they did well there. And here are sand dab growth plots. So largely they were similar across all sites, and if oxygen levels are driving growth, that may help explain it, because if you remember, oxygen levels were also very similar across all sites. What you'll notice in the last graph, however, is there is this trend towards less weight loss at the poor sites that were in the upper and middle slough. And because my experiments took place in 2 different seasons, we have to be cautious when drawing any kind of comparison, but I did want to just show you a side-by-side visual of their growth patterns or shrinkage patterns. So again, to reiterate, for the sole, we had the best growth at Salinas River, lowest at Kirby Park, and then intermediate at the fair sites. And then for sand dabs, on the left-hand side for the fair, you have a lot of shrinkage in body condition. This is showing you condition factor, which combines the previous length and weight metrics into one single metric of how fat the fish is. And then for the poor sites, they still lost condition, but it was significantly less than they did at the fair sites, and we'll talk about why that might be later on. So now for the principal component analysis. So this is basically taking all of the mini-dot calculations that I did and reducing them to 2 new uncorrelated principal components. I'll call them PC1 and PC2. So basically what I want you to see here for sole in August, principal component 1, which is that horizontal line, is going to be defined by higher temperature, lower oxygen, and then less time spent, or more time spent below the critical oxygen thresholds. And then principal component 2, which is the vertical axis, was a little bit weaker, but mostly defined by higher temperatures as well. And then when you plot PC1, PC2 against growth and survivorship, there's going to be a negative relationship for both components. And then, so just to remind you what each of those represents, this matches up with what we saw earlier with growth and survivorship, lower growth and survivorship for English sole being driven by the duration and concentration of hypoxic events, and also the maximum temperature. So 10th percentile DO and time spent below 4 milligrams per liter. And then to that end, a common theme that we've seen here is that temperature can really impact the health of fish. So it can be tricky in field experiments to pinpoint an exact threshold for survival for when that happens, but we noticed something interesting for English sole. So this is telling you where there's a change in slope and with maximum temperature versus survivorship of English sole. And that happens right around 21, 22 degrees, which actually matches up with lab studies that have recorded that as the upper limit of their thermal tolerance. And also based on the literature, I would have expected to see a similar pattern with sand dabs, but it didn't get hot enough in November to really verify that theory. And so for the sand dab PCAs, what you'll see is for the PC1, that's going to be associated with higher DO, lower maximum and average temperatures, and also lower time below hypoxia thresholds. And then PC2 is going to be associated both with lower average temperatures and actually lower DO concentration as well. So as would be expected, because the conditions weren't as extreme, there's less strong relationships than we saw with the English sole, but we still do see that survivorship is associated with the component that includes temperature. So that means it's going to go up when temperatures are lower. But then growth actually is associated positively with also lower temperatures, but lower DO. But again, because DO was so similar, that would lead us to believe that that relationship is more likely driven by temperature than difference in oxygen between those sites. Okay, so to recap there, English sole performed poorly at poor, salty, hypoxic sites in August, but performed well in the Old Salinas River channel. And then in November, sand dabs survived better at the cooler fair sites, but may have grown better at the poor sites. So at least for the English sole, that does match up with what we've seen from other studies. showing that oxygen and distance from the estuary mouth are driving nursery quality, and also that low DO, high temperature sites are bad for survivorship and growth. The Jeppesen study actually found that it was growth and survivorship were positively related to salinity, but those recordings didn't go above 34 PSU, so they weren't hypersaline like my sites were. So now let's talk about why we might be seeing some of these patterns. Why was temperature so important? Temperature is a controlling factor, so it's directly controlling the rate of metabolic processes. And in the wild, 76% of speckled sand dab distribution is described by temperature alone in King Harbor, California. And then when it gets too warm for fish, they start to show signs of stress, like increased heat shock proteins and cortisol production, and that in turn can decrease appetite, feeding efficiency, and then also reduce body condition as well. And then the warmer it is, the higher risk of bacterial infection, which can further put fish at risk. Now for oxygen, we saw at least for August that mortalities and weight loss for English sole were associated with sites characterized by hypoxia. Compared to other fish that spend a lot of time up in the water column, flatfish tend to be more resilient to hypoxia because they're not as active and they therefore don't have as great of oxygen requirements. So even at low levels, they still do pretty well. This is a picture of Juliana Cornett's study from a couple of years ago. So you see that she tested multiple different oxygen levels, but fish didn't really start trying to compensate until 2 milligrams per liter. So that means that the mortalities that I was seeing, even though they were related to low oxygen, was not likely directly due to hypoxia. And if temperature is a controlling factor, oxygen is what's known as a limiting factor, so it's not directly controlling the metabolism, but if you don't have enough of it, then you have to just worry about maintaining homeostasis, and then you don't have as much energy to invest in feeding, digestion, and metabolic function, so that's going to limit your growth as well. And then salinity, I didn't have continuous data like DO and temperature, but from the limited measurements that I did take, you could see that it got really salty in the slough, in the upper slough in August. And that's important because osmoregulation or maintaining your internal salt balance requires a lot of energy. Normal seawater is around 35 PSU. Anything higher than that is going to be considered hypersaline, and that can result indirectly in a reduction of growth because the fish has to spend energy on pumping the ions out and maintaining their salt balance. So in the upper slough, that may have been part of the reason why we saw a little bit lower growth, whereas in November, when the salinity gradients were reversed, that's also when growth was better in the lower slough. So previous studies show that 25 to 30 PSU is actually optimal for flatfish growth, and that's lower than full-strength seawater. I do want to point out that there are some other factors not quantified in this project that may have been important. So most notably, prey availability. There was a study in 2007 that did a benthic survey in Elkhorn Slough. I was focusing on more of the abiotic factors, but if I were to do this again, I'd like to take some core samples as well. But we learn a couple of things from this survey, and one is that as you move up the slough, richness decreases, but abundance increases. And my fish are both trophic generalists, so they'll eat anything. It doesn't really matter to them if biodiversity is high. And then the other thing we learn is that because the river channel is tucked away and it's protected from the erosion that affects the lower slough, the abundance of invertebrates here, it's lower than the upper slough, but it's actually significantly higher than the lower slough sites, Seal Bend, VR Beach, and the harbor site. And then physical factors like substrate, turbidity, and habitat complexity can play a part as well. Flatfish like to bury. The slough is at risk of a lot of erosion, so this is not my area of expertise, but over the years, as erosion has impacted the channel, it can wash away the sand and leave behind a lot of clay deposits, so that's going to be less ideal for the flatfish. And then any of the factors that we mentioned could exacerbate one another. So that also was not quantified, but timing matters as well. So if you recall, they're experiencing in the upper slough hypoxic stress at night when temperatures are cooler, and then during the day, it's really warm, so oxygen is fine, but it's too warm for them to function well, so they never really catch a break. Whereas in Salinas River Channel, they only had the one stressor, which was hypoxia at night. So that means during the day when oxygen levels were high, they were able to still feed and behave normally because temperatures were within their preferred range. As you've probably gathered by now, there are a number of challenges to this project. So first of all, field work by nature is messy, and estuaries themselves are complex. They're not static. They consist of a patchwork of water bodies with a bunch of different characteristics. So identifying driving factors of even a single parameter can be difficult. And then related to that, things like storms, potential animal interference with my cages, and cage loss, as well as a red tide that took place during one of my experiments resulted in some gaps in the data. And then glaringly, I was working with two species over 2 different months with completely different environmental conditions. And largely that was due to the logistical constraints of how long it took to clean, repair, and construct new cages. Took longer than I thought between experiments. So that's why they took place not during the same season. However, in spite of all of that, there is still a value to fieldwork. So I would suggest that future studies, I was focusing on getting multiple replicates at several different sites. I would suggest future studies might concentrate on fewer sites, but testing both of the fish at the same time. Lab experiments can offer valuable insight, but they also tend to underestimate the impact of the stressors that they're experiencing. So even with all of the challenges, the results of the study could have important implications. For one thing, the quality and quantity of nursery habitat. If that's reduced, that can have impacts for the adult offshore fishery. So this can result in a decline in those populations. So understanding what's happening in our nurseries could be valuable information for stock assessments. And then benthic fish also act as sentinels of environmental health for a system, and so the potential sensitivity of English sole and speckled sanddabs make them possible indicator species for evaluating water quality in Elkhorn Slough. And then this research also highlights the need for local long-term monitoring efforts that are already happening with Elkhorn Slough Foundation. So they're developing total maximum daily loads, which are water quality targets that can influence management. And some practices to reduce nutrient input into the watershed include specific crop rotation, planting cover crops, buffer zones. But also my results suggest that we also consider water quality beyond the nutrient lens. By definition, eutrophication is excess nutrients, but the symptoms of eutrophication, so fish kills, biodiversity loss, are largely not directly related to nutrient concentrations themselves. So the water quality report card that I was doing, DO, pH, and nutrients, salinity and temperature are both missing from those. But those can still have important implications. So to sum everything up, good nurseries may require relatively low water temperatures, low salinities, and high dissolved oxygen, and although not quantified here, also high prey availability. And then nursery habitat in Elkhorn Slough varies seasonally and spatially, and also may even reverse seasonally. And finally, this research potentially highlights some growing management concerns for the future of our fisheries. I have a lot of people to thank. So thank you, first of all, Scott, for everything from accepting me into the lab to going out in the field and providing feedback very promptly and a little push when I needed to actually give you something to provide feedback on. To that end, also, my committee members, Dr. Cheryl Logan and Tom Connolly, usually you hear about students waiting on their committee for feedback, but with us is the opposite. They were waiting on me to provide something that they could give feedback on. So thank you for helping with the planning the project, and also all of the input on the actual document. And then thank you to the Coast and Wave Award for funding. Helena Lindsay was crucial in providing guidance for acquiring funding in the first place and getting this project started. My fellow flatfish keepers, Juliana and Alex, and Kevin Hinterman, for all of your help in the aquarium room, and also just helpful feedback you've offered at every step of the way. All of the different people in the lab, Steve, who offered help with water quality and instrument calibration. Lots of field support. Daphne and Marcel, I relied on you as my boat drivers. Without you, I wouldn't have been able to access my sites in the first place. Everyone at Marine Ops for letting me use that as my 24-hour office. JD and Jackson, if I was ever struggling with a problem, you always jumped in to help. Brian rescued us from the slough on that day. And of course to my friends and family who've offered lots of support. These three individuals, Jesse, Kinsey, and Nick in particular, a lot of literal blood, sweat, and tears went into this project. So Jesse, you're my primary boat driver, and a lot of field days is just the two of us, a lot of early mornings and long days, so thank you for all of your help. Kinsey and Kevin, thank you for providing the snacks today, and also just always jumping in, literally, to help in the mud. This was my fault, but she was the one who took the anchor and like dragged it out so our boat wouldn't float away overnight. And then Nick, I would not have been able to build all those cages in time without you, so you put in a lot of hours of work there, and also just had really good ideas for how to make the construction process better and more efficient. So thank you to everybody. And now I can take some questions.
It does not project your voice, but it allows people on Zoom to hear the question. Who would like to ask Grace a question? Don't be shy. All right. We'll do Kevin first, then Nate.
Great job, Grace. It's cool to finally see this all come together. I have two questions. So one is at the end, you mentioned how those report cards don't include temperature and salinity.
Mm-hmm.
If you were to include temperature, because that seems like it's probably the most important one, how would you include it? Would you do maximum temperature that each site sees? Would you do average daily temperature range, or what do you think would be most useful for
They still collect data on temperature and salinity the same way they do with all of the other parameters, which is they go out to the sites and measure it once a month. Those just don't make it into the model. So how they grade it, they look through the literature to find out what is considered an acceptable point for the parameter, and then record how often it falls out of that range and how far. So I think they would do that just the same way that they quantify dissolved oxygen. So it would be whatever temperature the water is at the time that they get it, if it's within that range. But I think maximum temperature would probably be more useful in terms of quantifying nursery quality, but for that they would have to have continuous measurements instead of just going out once a month to sample it.
Yeah. My second question, I guess testing your memory. So you had high mortalities, especially with English sole, which is expected. Yeah. I was just kind of curious if you remember looking at some of the dead ones or some of the ones that weren't doing so well. Did you ever see like tumors or lesions on their bodies? Because we've been seeing that pop up quite a bit in captivity.
Oh, okay. Yeah, so they don't do well in captivity, as we know. I think the cage also could be considered like partial captivity. In terms of keeping them in-house in the aquarium room, I feel like that had to do with how small their mouths were, and they just weren't able to feed as well. I did not see any growths on them because when we pulled up the dead ones, by that time they were usually just in the skeleton. So they're either fully alive or fully dead, and I wouldn't have been able to see they were chewed at a little bit by the other fish. So yeah, they're dead.
Cool.
Good job, Grace. I was curious about the behavior of the fish and how it varies potentially between the two species. You mentioned that Hughes showed in the 2015 study that they seem to scurry away from hypoxic zones. And so I was curious about the degree to which either or both species can move vertically and whether that also affects the environmental conditions within the slough. Like, is there variability as a function of depth, and does their behavior reflect some of that?
Because they're a benthic species, even when they need oxygen, they don't have as easy of a job acquiring it. Like, some fish will do the surface gulping when they need to get oxygen, but pelagic fish operate in 3D space. Essentially, you can go up, down, and then laterally, but flatfish are like us because they're just at the bottom, and so they can basically move only back and forth. So I don't think that they'd really be able to find vertical refuge, but especially in the spots where they would need it are really shallow. So that's in Hudson's Landing. I don't think there really is that refuge, so they could only move downstream. And then once you get to the deeper sites by Seal Bend, they could move up and down, but they wouldn't need to because conditions are better there anyway.
We have noticed in captivity that the speckled sand dabs tend to come up to the surface much more readily than the English sole. And like Juliana found that in the lab experiments that they actually did decently well at pretty low oxygen, but she thinks that's because they were actually going up and circulating. So they probably have more capacity to do that. Maybe that's why they do a bit better at low oxygen.
Thank you.
Other questions? Who's got a question?
I had a question on that similar vein. I was curious if you thought that their ability to move might affect like the types of results you're seeing. So like you had them in cages, obviously, so they couldn't escape. Do you think that like they would be able to move at distances great enough to escape like high or low DO and high temperature, or do you think it's like no, they're kind of screwed if they're in the wrong spot?
I think based on other studies that have tracked movement, not necessarily in Elkhorn Slough, but other spots, they don't go very far, but the slough is small enough that I think they would have enough time to escape, because the temperature, even though it happens, daily, hourly flux, it's not going to be like immediate. I think it would be gradually warming, so they could have time to move down the slough would be my guess.
Question?
I remembered looking, I don't remember which slide it was, if it was the August or November, but it was like on day 10, when you're doing like the mortality over time. It seemed like day 10 there was like a large mortality event. Did you see in the data what might have caused that, if it was like O2 or?
Let's find, I don't, was that this one?
Yeah, that's maybe day 9.
So the days, these are done weekly. So if it was a specific day within the week, I don't know that. So if it's showing up day 10, that doesn't necessarily mean all of those mortalities happened on that day, but it was by the end of that experiment. So that could have happened at any time within the second week. So to my knowledge, there wasn't like an event that caused a bunch of deaths, but I would have guessed that those could have happened over the course of a couple of days. We had the red tide one for August. I didn't include those data because I think that was what caused a bunch of mortalities at the harbor site, which should have been more stable. But that was one kind of anomaly that could have caused higher mortality than expected.
If my voice works. Great job characterizing the differences in oceanographic conditions and physical conditions and ecological conditions across the slough. Given the number of ag fields around and the potential for pesticide pollution. I was just curious if you know much about like the concentrations of pesticides in the different parts of the slough, and like if you could kind of hand wave, like, do you think that they're having any impact on your data?
I unfortunately actually don't know too much about the pesticides, but just based on the fact that the poor sites were closer to the terrestrial input sources, that could have been a factor. I feel like the August data still makes a strong case for temperature being more of a driving factor. But yeah, I'm not sure about that. But then in the wetter season for sand dabs, when we have a lot of rain, that could increase the pesticides at the same time. So I'm not quite sure what impact that would have.
I'm going to ask a question. So, I mean, your results suggest that about half of the slough is no longer, decent nursery habitat for these fish. So do you think the remaining parts of the slough is there enough area there to basically continue supporting all the little baby flatfish, or have we lost some general nursery function due to these environmental changes?
It's such a big portion of the slough that wasn't good, so I don't know the math in terms of recruitment and then what that means for adult populations, but I would say there's strong evidence that we're losing some of it. There is a study that did long-term data of temperature in the slough, and they said that between 1988 and like 2003, I think the average temperature in Hudson's Landing in July went from 18 degrees to 23 degrees or something like that. And so that, in just that short span of time, to have that much of an increase means that we lost a lot of potential habitat since the 70s and 80s at least.
Yeah, and the fish used to go all the way to Kirby Park, but it's
that used to be their favorite spot.
Now they don't do so well there, and that's where all the prey is too, right? So yeah.
Where all the what?
That's where a lot more
Oh,
prey.
yes,
potential prey
yeah.
are. Okay, any other final questions? Ooh, one more. Last committee member.
Great job. So I just wanted to follow along with Scott's point that the Hughes paper you were showing was showing a lot of interannual variation with the PDO and climate factors. Do you know what was happening with the PDO or other interannual climate variability at the time this study was done?
For my year, I don't. I'm sorry, I don't know. I think sometimes the report card tells you like rain notes for that year, but I don't think it had them for that year. Do you know if 21 was in El Nino?
23 was kind of El Nino, super rainy. I forget what 21 was.
Yeah, unfortunately, sorry, I don't know the answer to that one. But I know the ENSO cycles do drive those patterns a little bit. But so I'd be interested in seeing this kind of experiment repeated over different years and different months to get a better idea of what's going on.
So Grace's committee, we're going to take her in the back room and have a short little committee meeting. But let's congratulate her, and then she'll come celebrate with everyone.